Hello friends, welcome to TechQuest channel. This is for the medical technologist by your tech. In this video, we will discuss about bacteriology, sample processing and about direct smear for staining and selection of culture media. All appropriate microbiology samples should have a direct microscopic examination. The direct examination serves several purposes. First, the quality of sample can be assessed. For example, sputum sample can be rejected that represents saliva and not lower respiratory tract secretions by quantification of WBCs or squamous epithelial cells present in the specimen. Second, the microbiologist and clinician can be given an early indication of what may be wrong with the patient. For example, CSF direct smear and Albert stain report with suspected Corynebacterium diphtheriae from throat swab can give an indication for the clinicians. Third, the workup of specimen can be guided by comparing what's grown in the culture to what was seen in the smear. When 3 types of organisms are seen in the smear and only 2 types are grown in the media, third could be an anaerobic organisms, need special environment for their growth. Fourth, the direct smear also help us to decide the culture media to inoculate the sample to additional special media for their growth or to select the size of the media for isolation. The most common stain in bacteriology is the gram stain that helps visualize rods, cocci, WBCs, RBCs, or squamous epithelial cells present in the sample. The most common direct acid-fast stains are aramine, rhodamine, zilnacin, and quinone stains. How to make a smear? Generally, specimens are placed on a slide using a swab containing patient materials. To avoid contamination of culture media, once the swab has touched the surface of the non-sterile slide, it should not be used for subsequently inoculating media. The swab to be rolled onto a glass slide and just not rubbing. For liquid samples using a sterile pipette, the specimen can be aspirated and placed on a glass slide. Use a new glass slide, write the specimen number and write a circle on the bottom side or other side of the slide. Make a smear on the front side so that it is easy to focus the smear after staining. Let's see the list of sample and the direct smear methods and the common media used for each sample. Aqueous or vitreous fluid, gram stain is important. Blood agar, chocolate agar, McConkey agar, Sabrot's dextrose agar, thioglycolate medium are commonly used. Abscesses, also wound lesions, ulcer. If it is superficial, gram stain is done. And again, blood agar, chocolate agar, and McConkey's agar is used for inoculation. If deep abscess or pus, gram stain is done. Blood agar, chocolate agar, and McConkey agar, and any anaerobic media is used for anaerobic bacterial identification. Body fluids, amniotic, abdominal, peritoneal, ascites, bile, synovial fluid, pericardial fluid, pleural fluid. We can use anaerobic and aerobic blood culture bottles. Gram stain is done for all these samples. Blood agar, chocolate agar, McConkey's agar, and thioglycolate medium are used for inoculation. For bone, the gram stain is done. Blood agar, chocolate agar, McConkey agar, and thioglycolate medium are used for inoculation. CSF, cerebral spinal fluid, gram stain after centrifugation at 1000 to 1500 rpm for 10 minutes, or the cytospin smears can be used for gram stain. Blood agar, chocolate agar are commonly used. We can add thioglycolate medium for CSF collected from shunt. For ear, inner ear, gram stain is done. Blood agar, chocolate agar, McConkey's agar, and we can add thio if prior antimicrobial therapy is done. add anaerobic culture plates for tympanocentesis specimens. Outer ear gram stain is done. Blood agar, chocolate agar, and McConkey's agar are used for inoculation. From eye samples, conjunctiva gram stain, acridine orange stain, histological stains like Giemsa stains are used. Corneal scrapings, gram stain, acridine orange stain, blood agar, chocolate agar, Savarod's dextrose agar, thioglycolate medium, and anaerobic media also used for inoculation. From gastrointestinal tract, gastric aspirate, gram stain is done. Acridine orange stain, blood agar, chocolate agar, McConkey's agar are used. The Columbia agar with colistin and nalidixic acid also used for gastric aspirate samples. Rectal swab. Normally direct smear is not done. Methylene blue stain may be done for fecal leukocytes. Blood agar, McConkey agar, XLD or selenite or selective Campylobacter agar can be used. Stool culture, methylene blue for fecal leukocytes, blood agar, McConkey agar, XLD, and selective Campylobacter agar is used. Cervical swabs, gram stain is done normally. Blood agar, chocolate agar, McConkey agar, Thayer-Martin medium, endometrium samples, gram stain is done. Blood agar, chocolate agar, McConkey agar, Thayer-Martin medium, and anaerobic medium can be used. Urethral swab, gram stain, blood agar, chocolate agar, Thayer-Martin agar are used. Vaginal swab, gram stain for bacterial vaginosis, especially WBC, gram-positive rods indicative of lactobacillus, and curved gram-negative rods indicative of Mobilingus species. For prostate swabs, gram stain is normally done. Again, blood agar, chocolate agar, McConkey agar, Thayer-Martin agar, Columbia agar with colistin and nalidixic acid are used. Respiratory tract, bronchoalveolar lavage, gram stain, and other special stains as requested. Example, Legionella DFA, direct fluorescent antibody stain, and acid fast stain are normally done. The plates are blood agar, chocolate agar, and McConkey agar. Sputum or tracheal aspirate, gram stain, and other special stains as requested. Example, Legionella direct fluorescent antibody stain, acid fast stains are normally used. The plates are blood agar, chocolate agar, and McConkey agar. Nasopharynx swab, gram stain, blood agar, chocolate agar, add special media for Corynebacterium diphtheriae, pertussis, chlamydia, and mycoplasma. All blood stain and acid fast staining if requested. Pharynx, gram stain, blood agar, add special media for Corynebacterium diphtheriae, Neisseria, and epiglottis haemophilus influenza. Add all blood stain, methylene blue stain if required. Tissue. Sample may need to be homogenized. Gram stain, blood agar, chocolate agar, McConkey agar, anaerobic media, thioglycolate media if indicated. Urine sample, clean, voided, midstream urine, gram stain, blood agar, McConkey agar, or cled agar may be used. Plates for urine colony count should be done. Straight catheter, gram stain, plate quantitatively at 1 in 100 and 1 in 1000 for colony count, blood agar, McConkey agar, or cled. Indwelling catheters, gram stain, plate quantitatively at 1 in 100 and 1 in 1000 for colony count, blood agar, McConkey agar, or cled can be used. Suprapubic aspiration of urine, the gram stain, plate quantitatively at 1 in 100 and 1 in 1000 for colony count, blood agar, McConkey agar, or cled, and anaerobic media is added. Foreign bodies, IUD, plate as soon as the sample is received. No staining is required. Thioglycolate medium is enough for the growth. IV catheters, pins, prosthetic valves, no staining method, blood agar, and thioglycolate medium. Selection of media to inoculate for any given specimen is usually based on the organism most likely to be involved in the disease process. For example, CSF specimen, you must consider the most likely pathogens that cause meningitis. They are Streptococcus pneumoniae, Haemophilus influenza, Neisseria meningitidis, Escherichia coli, or Group B Streptococcus, and select the media that will support the growth of these organisms. Blood agar and chocolate agar are a minimum. Likewise, if a specimen is collected from a source likely to be contaminated with normal flora, we need to add a special selective medium to suppress the normal flora and allow the pathogens to be recovered. Thank you. Please like, share, and subscribe. Thanks.